Now we will move to Jennifer for her presentation on human health.
Good morning, everyone. My name is Jennifer, and today I'll be talking about the effect of extracellular matrix and signaling protein on self-assembly of liver biliary cells in hyaluronan hydrogels. So to start off, a bit of background. So liver organoids are currently critically needed for drug discovery and transplantation purposes. And if you take a look at the liver in the middle, all those green branches are actually bioducts. And bioducts are essential for hepatocyte survival. So without bioducts, the hepatocytes or the liver cells are not going to survive for too long. However, the problem with the current liver organoids is that they don't have any bioducts. And they often use a mouse tumor-derived biomaterial, which means that it is unsuitable for transplantation. So currently, to modulate the liver in 2D, people typically use JAK1 induced notch signaling, or they sometimes also manipulate the composition of the 3D microenvironment. So for example, this is a study previously done in our lab, and these are mouse cholangiocytes. So the cholangiocytes are the cells that make up the bioducts. So the image on the right does not have an JAK1 in it, and then the image on the right has JAK1. And then both of these images are stained for SOX9 and CK19, which are both cholangiocyte markers. And then the image on the right obviously had like significantly elevated amount of SOX9 and CK19, which tells us that the JAK1 can promote cholangiocyte differentiation in 2D. And in terms of the extracellular matrix, laminin is a major protein. So this is also a study done in 2019, not from our lab, but from a collaborator's lab. These are also cholangiocytes. So they're also trying to grow bioducts in culture. And then the image on the left does not have any laminin in it, and then the image in the middle does have laminin. And then this, like the image on the middle does have a circular structure that resembles the cross section of the bioduct, as you can see on the right. So this tells us that the absence of laminin can lead to defective bioducts. So now we know what's happening in 2D. So we know that in 2D, JAK1 is very important. In 2D, laminin is also very important, but we don't know what's happening in 3D. So that's where our study comes in. We're trying to find out what's actually going on in 3D. So we hypothesized that the ECM proteins, more specifically collagen and laminin, and JAK1 can modulate the growth and cellular self-assembly of cholangiocytes in 3D HA-based hydrogels. In order to make this gel, we have our HA ketone and HA aldehyde, as you can see in the image, and we also have various concentrations of laminin and collagen. And here we used pacoxamine as a cross-linker, and we also, of course, have our cells, our mouse cholangiocytes. And then in order to add JAK1, we also immobilized JAK1 in our gel on the HA ketone and HA aldehyde. So after we have our gel, like after we encapsulated our mouse cholangiocytes in our gel, we let it sit for 90 minutes. And then this is the gelation process. So after this 90 minutes, the gel was solidified. And then after that, we add media into it and after that we add media, and then we let it culture for up until 6 days. And at day 1 and day 6, we perform confocal imaging on it, and we also do a pressed blue growth assay to tell us how many cells are actually in it. This is our data from the pressed blue assay. So we basically measure the amount of cells in day 1, and we measure the amount of cells in day 6. And then to calculate the cell proliferation rate, we use the amount of cells in day 6 to divide by the number of cells in day 1. In other words, the day 6 data would normalize to the day 1 data. And then that's how we generated these graphs right here. So the y-axis is the calculated cholangiocyte proliferation rate, and then the x-axis here are just gels with different collagen concentrations. So the 4 pink bars on the left of each graph actually does not have any laminin in it. And then the four green bars in the middle of each graph does have laminin. And then the red bar the red bar on the furthest right is matrigel, which is the previously mentioned mouse derived the mouse tumor derived biomaterial, which is the current gold standard like in the field. But so there's this general trend here that all the green bars are higher than all the so yeah, there's a trend here that all the green bars are taller than all the pink bars, which suggests that laminin improves the mouse primary cholangiocyte proliferation. And after we put these graphs together, we generated this graph. So the black bars are HA hydrogels with JAK1 immobilized in it, and then the green bars does not have any JAK1. It's just a simple HA hydrogel. And we can also see that all the green bars are taller than the black bars, which tells us that cells can grow slower in JAK1 HA hydrogels in comparison to the simple HA hydrogels. So after we studied the proliferation and cell growth of cholangiocytes, we took immunofluorescent images to study the self-assembly. So these images here are stained for actin. So the greens are actin, and then the blues are just the nuclei. The image on the left does not have any JAK1, and then the image on the right does have JAK1. And so the middle image. The middle image does have the structures, so which resembles the bioduct structures that's shown on the right. However, the exact conditions are confidential at this stage, are not to be and are not allowed to be disclosed. But from this image, we can see that the self-assembly of cholangiocytes can be observed with appropriate signaling and appropriate gel composition. So in conclusion, so from our Presto Blue assay, we know that JAK1 does not promote proliferation of cholangiocytes in 3D, and we also know that the inclusion of laminin promotes cell proliferation. And from the immunofluorescent images, we can see that self-assembly of cholangiocytes can be observed with appropriate signaling and with appropriate gel composition. In the future, we're trying to test out different signaling proteins to see and also to see the combined effect of different signaling proteins, and we also want to evaluate the differentiation of cholangiocytes. So the overall goal of this project is that we want to ultimately develop a reproducible liver biliary organoid for drug discovery and transplantation purposes. And last but not least, I want to thank my professor, Dr. Molly Shokit, as well as my mentor, Dr. Rizwan, and the PhD student Chris, as well as another summer student Tracy, and everybody in the Shokit lab, of course. And I welcome any questions.
Hi, Jennifer. This is Solmaz. I have a quick question about your 3D confocal imaging. You mentioned that you took pictures day 1 and day 6, if I'm not mistaken. So which day is that, these figures?
Oh, yeah, these ones were taken at day 6, I think.
Day 6? So can you show me where the cells are, and then how do you know if they proliferated?
Oh, so yeah, so thanks for the question. So the question is regarding how we know they're differentiated. So in this case, we actually don't know if they're differentiated because we didn't stain for any differentiation marker. So say like SOX9 or CK19. In this case, we only stain for DAPI. So these green dots here, these blue dots here, these are the nuclei. So that's how we can tell where the cells are. And then we also stain for actin, which is a like cytoskeleton a protein, which just tells us how the cells look like. So just a marker for cell morphology, but we couldn't tell what kind of cells they are from these markers. And that's why that in the future, we would like to evaluate the differentiation.
Okay, thank you so much. Thank you.
Yeah.
Thank you for your presentation, Jennifer.